All right, welcome everyone as you're coming in to our DiMe Journal Club, Advancing CNS Care with Digital Measures of Gait. While people trickle in, I'm going to already go through a couple of quick housekeeping rules so that we have that out of the way. Today's session is going to be recorded. There is going to be a Q&A session. If you have a question, you have two ways to ask that question. You can raise your hand, and in the background, somebody will take you off mute so you can ask your question live. Or if you prefer, you can type it straight into the chat box, and I will do my best to bring it into the conversation. I'm just checking here. Looks like we've got a decent group of people who came in. So we can get started. Let me just do like a brief introduction of the speakers. And of course, for everyone here who is going to be speaking on this work, feel free to, when your section comes up, to give a bit of more elaborate background if that's relevant. So we are joined by Melissa and Brett from the metadata team. Melissa is the VP Engineering and Biomarker Analytics at Metadata. Brett is a Senior Data Scientist at Metadata. And then we have Professor Reed Gerchek and Ryan McGinnis from Clemson University and Wake Forest University School of Medicine, who are authors on the work that we're going to be discussing here. And my name is Ben Van Adries. I'm the Chief Delivery Officer at the
Hey,
Digital
Tucker,
Medicine Society.
come here.
To
Lie down.
I hope that wasn't directed at me because I heard somebody say lie down. The to kick us off, I'm very quickly going to go over three DiMe resources, kind of quick fire style, that directly relate to the work that we're going to be discussing here that Metadata and their collaborators have been working on. So a lot of you may be familiar with the V3 Plus framework, which was originally launched back in 2019 and has had a really outsized impact on how we're validating digital health technologies and how we're setting up those studies.
I
And
can only
we
see a
really
Zoom window. Just there we go.
the slides are showing.
Yeah, go ahead.
All right, thank you. Um, and was updated last year to include usability validation as well. So again, very briefly, for those of you who are maybe less familiar, the framework really splits the work that needs to be done to evaluate a sensor-based digital health technology into multiple stages. There's verification, which is focused on verifying the sensor performance, so the hardware side of things. There's usability validation that evaluates if the DHT can accomplish specific goals with ease, efficiency, user satisfaction. Analytical validation is something that we're going to be talking about a lot in this session because it pertains to evaluation of algorithms and their performance. And then clinical validation, last but not least, is evaluating if a digital health technology can identify measures measure, predict a meaningful clinical, biological, physical, functional state or experience in the, and this is important, stated context of use, right? And this framework is modular in the sense that as you are, fiddling with your digital health technology, as you're trying to add more features, as you're trying to add more performant algorithms, as you're trying to expand to other patient populations, you can just kind of slot back into these various stages and update that piece of it without needing to redo all of it, depending on your technology. So this is a critical resource when doing anything with a sensor-based digital health technology to ensure that as a field, we're developing technologies that are fit for purpose. And one of these components I mentioned already, analytical validation, is something that we're going to be seeing a lot indirectly in the work that's going to be presented afterwards, is often one of the more tricky ones because it relies on defining a good reference measure. And a good reference measure sometimes is easy to come by. Sometimes there's a so-called gold standard, gold clinical standard that can be used to compare something new to. But often, and especially for more novel measures, that is not the case. And so the validating novel digital clinical measures work, which is a bit of a mouthful, sets resources that allow you to really break apart the various stages of an analytical validation study and helps you identify the most rigorous reference measure that is currently available. So that's a highly valuable resource too. And then last but not least, we have developed a core set of core measures of physical activity, which is a set of core measures that across a broad set of therapeutic areas ensures that there's more alignment in the field, and it also increases our ability to compare data from multiple studies across multiple studies, right? And that includes obviously measures of gait, and it includes measures of postural sway, both of which will be discussed in the work that is upcoming. Like always, all of these resources are freely available on our DiMe website. And with that, I'm going to hand the mic over to Melissa to intro some of the scientific work we're going to be talking about here.
Great. Thanks, Ben. And thank you, everyone, for joining today. It's really exciting for us to be able to share our work with you. So Brett and I, as Ben mentioned, are at Medidata, and we've been working on digital biomarkers for quite some time. And then my colleague Ryan and Reed, we've also been closely working together. And the last big project we did together was in Huntington's. And the paper that we wrote is about to be published. So very timely for us to get back together today. And Ben, you can go to the next slide. As Ben said, we at Medidata are really studying and focused in the CNS space. And the reason why, we're spending a lot of our time and investment in this space is that it is a complex and a very burdensome field, both burden on the patients. There's 3 billion patients worldwide that are impacted by a CNS condition, and also the burden to our patients and to our sponsors that are trying to come up with new innovations and drugs and therapies in this space. So just as an example, in Alzheimer's, an average phase 3 study cost is $370 million, and the timeline is, approaching 8 years. So a big burden in terms of cost and time. And then at the end, there's a very high failure rate, so 95% failure rate. And on the next slide, we kind of take a step back and now looking at it in a different perspective around patient centricity. And these numbers here aren't specifically for CNS, but global trials overall really have some challenges retaining patients. So 85% of clinical trials don't retain enough patients. There's a big dropout rate. And there could be sites that have gone through all of the training and all of the onboarding and don't get to enroll a single patient, which is really debilitating for our work and for our field. And that is why we've really thought about the patient and site first. And on the next slide, the reason why we wanted to focus today on our expertise and some of the innovations that we're bringing forth on the biomarker side, specifically in CNS and specifically in movement disorders, is because we believe that this can be the tipping point of how we can design studies that are more patient-centric and really thinking about the patient first and meeting the patient where they're at. And so the platform that we're working on at Medidata really takes into consideration what is going to be compatible with the patient and their experience, and then how are sites best equipped to be able to meet those patients where they're at. And one of the things that we've been seeing over the course of our research in the digital biomarker space is that we have an opportunity with technology to really do these two things, to bring technology to patients, to be able to follow them and monitor them during their real world everyday lives and get a higher value of data back to the study team. And so Brett, I'm going to hand it over to you now to talk about our first example of how we are innovating. This is an example in multiple sclerosis, but really looking at postural sway and how we're monitoring patients in the real world at home and how we're getting a a lot of interesting data from these wearable sensors.
Thanks for that intro, Melissa. I actually do want to take a step back really quick. I do assume that people in a DiMe call are familiar with the benefits and why we like to use wearable sensors, but if not, I just want to give a quick intro as to why, right? Why would sensors even help a trial, right? If it's expensive already, what does a sensor cost money, right? Um, so in general, things done on paper or things of how do you feel today, 1 through 10, right? That's not very sensitive. Or and we are I'm sure most of us are familiar. That's how a lot of clinical skills are done as well. So really, the goal of sensors is to be able to detect a change in maybe someone's getting worse, or maybe they have a treatment and it's successful, right? We want to be able to detect those differences objectively. And the goal of sensors is to be able to do that in a very sensitive way, which then lets us reduce our sample size, which, right, less amount of people means you can have a shorter trial, less people you need to recruit. All of that saves money and gets drugs to the market faster. So I just wanted to back that up and say, okay, how do sensors help this? And then now we'll get into, okay, well, now that we kind of understand that sensors can be used to help, right, take these objective measurements that let us really get on that signal of if a drug works or not earlier, well, how do we implement them into a trial or into a study? And as Melissa said, the first one we're going to talk about, one of the first things we did was how do we actually do this for postural sway, which is really just standing balance. Then if you want to go to the next slide. So the first step we did is just see if it worked from the chest. So many of you may be familiar. There's a paper that came out in 2012, so it's quite well seasoned. It was looking at postural sway from a sacral sensor, and they compared it to a force plate, and that was great, right? We from there we all went forward and ran with it, and it was great. So basically, our goal was, can we compare to that paper? Do we get similar results? So for this, we had 16 people with MS perform they did I believe it was a 2-minute standing test on a force plate. So the force plate here is our ground truth. It's mounted in the floor. Stand on it, we know exactly what's happening. And then we basically just computed each of these postural sway features that was derived from that original paper from the sacrum. And what we found is we got very similar results to that paper. While the units are not the same, right? Being an accelerometer reported in meters per second and a force plate right, it reports it in meters. The correlations established in that paper are very similar. So we said, yes, okay, it works, move on. So that was our first step, is like, okay, does it do what we want it to do? Next, we want to see, is it useful? So Ben, can you go to the next slide? So, right, not only do we want to see if it was useful, we also wanted to know, can we do this better? And what do I mean by do this better? So I'll take a moment to explain what we typically mean by assessing postural sway. And it's in postural sway is something that we basically, it's a way to measure balance. And it's part of different tasks, like a Berg balance assessment, right? Where we will stand there and look at how much someone moves. But typically it's a 30-second standing bout where I just stand there, and I could be on a force plate, I could have a sensor, and I get one observation for, okay, how much did my torso move? How much did I move? Well, I mean, you're not just standing there for one period of time. So our thought here was, what if we just extended that time and built a distribution, right? So that's what you're seeing here in this figure. In that middle panel is, instead of just one observation, we actually we have a sliding window, and we compute that same feature for, say, I mean, it's been a little bit. You can see the citation on this was 2023. I don't remember exactly how many windows are there, but there's several, and what we end up getting is a distribution. And then on the right, you can see, right, for this particular example, that the 30-second standard single observation is not really representative of this. So to do this, we used a population of 39 people with MS, 21 had history of falls, and 12 were male. And they were also performed an EDSS from a neurologist. And then the patients completed a ABC, which is a balanced confidence assessment. They did a walking impairment survey and a fatigue survey. Let me go to the next slide. So, I mean, what we see is very exciting, right? Not only did we first show that we can measure postural sway from the chest, we then showed, look, we have this new way of computing it, and it actually gives us much stronger relationships. So what we see is that the distributions greatly increase the strength of the relationships we find into our clinical outcomes. And just right here, give you some nice concrete examples of this. When we looked in this data set, remember I said there was 39 people. I believe there was 21 that had fall history. If we looked at that single observation, all those features we generated from single observation, we were not able to distinguish any of them as a faller or not faller, right? There's nothing significantly different between the two features. But if we looked at the features we were able to generate from the distributions, we were able to distinguish between several features, the difference between fallers and non-fallers. And you may say, okay, well, you're just generating a lot of features. I'm sure a lot of them are terrible, right? Well, definitely there's going to be some less meaningful parts of the distribution. But what we actually find is that not only when we look at correlations to things like ABC and EDSS, we see that there's more significant correlations, but also on average they're stronger. So right, we're not just adding a lot of noise to it. And some concrete examples and what this figure is here. This is a ridgeline plot where we're actually seeing the postural sway parameter jerk on the x-axis, and then the figure on the left is EDSS. The figure on the right is ABC, and it's just sorted in terms of the patient value. And the strongest correlation in both of these was obtained with the 5th percentile. But what we see on the left is the correlation goes from, with a single observation, and that we go from 0.39 to 0.47, right? So you're gaining quite a bit there. And then on the right, it goes from 0.49 with a single observation to 0.54. So really, what we're gaining is an additional sensitivity. So we are able to detect differences even stronger, or detect differences in a better way than, which again, with doing techniques like this, allows us to have a smaller standard deviation in our assessments, and then we can, in theory, we don't need as much data to do this. And then now I think I'm going to pass it to Reed, who's going to talk about how we started to do this from the chest, or for gait.
Thanks, Brett. Yeah, so this is a very new study. These results were presented actually this summer in Copenhagen at an IEEE EMBS conference, and the proceedings have not yet been published online. But our goal in this study was to develop a method for characterizing the spatiotemporal nature of gait, things like stride time and stride length, using chest-worn accelerometer data. And to our knowledge, there was only one other package available for doing this, the Scikit Digital Health, or SKDH, but itself was a simple adaptation from an algorithm that was optimized for use on a sacrum or lumbar location. And we wanted to develop a method that was designed for the chest location from the beginning, which is an attractive location for several reasons, including wearability, sensitivity. It would be sensitive to bilateral gait motion, and so there's a potential to characterize interlimb gait asymmetries. And because it's an ideal location to measure cardiac activity. And so it would naturally permit such an analysis by augmenting the hardware with electrodes. So we can advance to the next slide. So at a high level, we use a series of filters and wavelet processing to identify first foot contact events, and this permits calculating, for example, stride time and other temporal metrics. And then the data from individual strides is used as input to a deep learning model to estimate stride length. So that's the pipeline at a high level. We validated the approach on 14 people, including 10 healthy older adults and 4 persons with Huntington's disease. Each person wore a biostamp sensor on their chest that recorded accelerometer data, and we collected marker position data using traditional marker-based motion capture during several overground walking trials over a 20-foot walkway. So on average, there was data available from about 9 trials for each person. And importantly, our algorithm operates almost exclusively on the magnitude of the accelerometer signal. And this means that a sensor to segment calibration would be unnecessary. However, we did need to calibrate the sensor to segment model only to distinguish a right stride from a left stride. So if you want to go to the next slide, please. In this study, we used a standard static calibration procedure, but we have developed an automated method for remote monitoring in practice, and we'd be happy to discuss that later. Now, the algorithm works by first filtering the accelerometer magnitude using a peak enhancement technique that's been described before by Soltani et al. In a previous paper.
And then heel contact events, those are the blue dots shown in the figure on the right. Those from Vicon motion capture, those were shown to align closely with the zero crossings of the wavelet transform signal. And those are the orange dots in the figure. Similarly, the foot off events occur in the valleys of that same transform signal. So we optimized the wavelet transform parameters using a grid search, and we distinguished left from right strides using the mediolateral accelerometer signal. Next slide, please. The ground truth gait events were identified using the Vicon measured foot marker position data, and we compared stride time, stance time, the percent stance phase, also called duty factor, and double support time to the marker-based motion capture method. We had data available from about 126 total walking trials for comparison, and the stride time results are shown in the upper left figure on the screen, and then stance time is being shown in the upper right. And you can see we observed strong correlations with the motion capture ground truth and root mean square errors between 43 and 67 milliseconds, at least for these two variables. And this was shown to be an improvement over the performance of the SKDH algorithm. Those results are being shown in the bottom row of figures. Next slide, please. So that was for the temporal metrics. For stride length, we used a deep learning model where the inputs to the model are the acceleration magnitude data during an entire and individual stride. We leveraged pre-training and transfer learning on an open source data set from persons with multiple sclerosis to overcome limitations of our sample size. So the pipeline and framework are being shown in this figure here. In this case, a deep learning model was initially underfit on our own data. It was then used to generate more stride data from the larger MS data set, and then the final model was initialized by pre-training on that generated data. We compared this approach to an algorithm based on an inverted pendulum gait model, which was described again by that Sultani et al. Group in the 2021 paper showed a moment ago. We used leave one subject out cross-validation to evaluate the approach, and again, the ground truth data were obtained from the marker-based motion capture. We found stride length estimation errors were within one standard deviation of the observed data. The root mean square errors were about 11 centimeters, and this was comparable and only negligibly better than the inverted pendulum approach, which is about 13 centimeters root mean square error. But the deep learning approach is attractive because it doesn't require patient-specific measurements like leg length and things like that, which are inputs to the inverted pendulum model. And our hope is that the pipeline altogether will facilitate new research opportunities, new clinical opportunities for free living gait analysis while leveraging the advantages of a chest-worn sensor. So we'll go to the next slide, and I'll turn it over to Ryan for our last paper.
Great, thanks, Reed. So I'm here to talk to you a little bit today about what happens when you try to take these measures that we've been evaluating in laboratory contexts and deploy them during everyday life, and all the sort of unknowns and challenges that go along with that. And we'll be talking about this particularly in the context of measuring postural sway remotely. Next slide, please. Great. So in this pipeline for analyzing postural sway remotely, we take data from two biostamp sensors, one that's affixed to the chest, one that's affixed to the anterior aspect of the thigh. We take data from those sensors, we feed them through a deep learning-based activity classification model that detects every time the participant engages in sort of daily activities like standing or walking, sitting or lying down. We can take those observations from the deep learning model and aggregate them into bouts of standing. And then for every standing bout that we identify, we can extract measures of postural sway. So just like you have a postural sway test in the lab, it may be 30 seconds, it may be a minute, it may be a minute and a half. You can create surrogates of that postural sway assessment at home as well. So every time a person stands during daily life, you get these measures of postural sway. The challenge with those is that these are often very noisy measurements. And so as you start to look at those data remotely, there needs to be some way to cut through that noise. So the data set we've done this in is the same sample that Brett described previously, 33 people with multiple sclerosis, roughly even split between folks who have fallen in the past and those who have not. So you could think of half the data set as being at high risk for experiencing a fall, half as being at low risk. And we wore these sensors at home for 48 hours. And as Brett mentioned previously, we have a clinical assessment, the expanded disability status scale, and then also patient-reported outcomes of the activity-specific balanced confidence scale, MSWS, and the MFAS, which is a measure of fatigue. Next slide, please. One of the primary findings here, and folks that have tried to analyze biomechanical data remotely sort of recognize this as a universal fact, but the data that we capture in the clinic looks very different from the data that we capture at home. The plot that you see here shows the z-score of differences for each of those postural sway features derived from all of the remote data and also from the lab. You can see large differences between the lab and that remote data. We've also done some unsupervised learning on these data to try to cut through some of the noise that exists in those postural sway assessments at home. And we identified 4 clusters in those data. And each of those clusters sort of are different along different postural sway features. Next slide, please. I think importantly, while those clusters are showing differences in their postural sway features, those differences really seem to matter. If you look at relationships between fall status and data within each of those clusters, or between those clusters and activity-specific balanced confidence scale or the EDSS, you see that those relationships are different. The second cluster happens to show the best performance at discriminating fall risk. They have an F1 score of 0.71. The second cluster also showed the strongest correlation to ABC and the strongest correlation to the EDSS. So I think as a practitioner who's looking at these data collected remotely, it's important to consider the context of the data and the context under which it's collected, and that all measurements of a given parameter may not be able to be interpreted in the same way. And in our case, it seems like data from one of these clusters is potentially much more meaningful for characterizing these constructs of interest in people with MS than data from the other 3. And so thinking through how we make those discriminations and choose which data to focus on is likely really important. Next slide, please. And that is it for my section. Thank you very much.
Thank you, Ryan and Melissa, Brett, and Reed, for that overview. I think it's a great way to get kind of a peek into recent advances in signal processing as it pertains to gait and postural sway, and I think especially kind of the diligent work that it takes to advance these techniques from the very early stage lab work, theoretical work, to in-clinic validation, and then last but not least, Ryan, as you were alluding to, the home validation piece, that also is so important here to make sure that measures are actually and algorithms are fit for purpose, and they continue to show the performance that that we need depending on the clinical case that we're trying to tackle. I think, like, if I try to summarize it for myself, it's really critical that, as a field, we continue to work on setting up these validation pipelines that work towards developing measures that matter, right? Measures of health that matter to patients. But that's an easy thing to say. And I think we had an interesting peek under the hood here of like how detailed and in-depth it goes when you are actually in the weeds of that process, trying to figure out what to use as a reference measure, how to validate, what to compare against, what conditions to cover for, what techniques to use, and so forth. So thank you again all for this overview. I would like to ask the audience to please not be shy, come off mute, join the conversation, drop whatever question you may have in the chat. When it ends up there, I will do my best to fold it into the conversation. But then I invite everyone, all of the speakers, to also come off mute, so we can have a discussion about what we just did, that we just saw. And while the audience is thinking of any burning questions that they may have, maybe to kick us off, question for the speakers is on calibration, which, kind of a boring topic often, but nevertheless critically important, especially when we're dealing with inertial measurements like accelerometers and so forth, where if you're looking at how things are done in a clinic, a very controlled environment, very controlled protocols, and then you need to take that out of the clinic and bring it to a home environment, you typically, I'm assuming things here, will need some way to either calibrate your device and or set people up for a specific exercise that provides a context of measurement that will be easier to interpret afterwards. So I would welcome comments from the group on how you're dealing with that, things that you've seen work in the past, things that you've not seen work, things to pitfalls that are very common and so forth.
Yeah, happy to jump in there on this one. So I'll just start to say we have absolutely done just asking participants to do manual activities, and that works, right? But we have heard and learned and seen the data that it also doesn't, and it doesn't work because some people will tell you they did a standing test and they clearly didn't, or they just either don't do it, or they say they did it but they didn't do that activity, right? We've had a lot of obviously a lot of good data come through, right? Because overall you can still do the work, but there are definitely plenty of examples of where it's just like it's clearly not something people love to do, and it's something that we would like to move past. So that's we are using a process we're calling it like auto calibration, where we're calibrating from the data we find within the sensor. So what we're actually using we're using a orientation sorry, oh, no, we're using a gait detection model that doesn't require any orientation adjustments. We're just using the magnitude, and we're finding the longest gait bout because we can assume the person to be upright. And then we're using that period to then rotate our sensor. And then from there, we use some more wavelets and principal components to then align the sensor. So basically, if the person wears the sensor long enough to walk, I believe I consider anything that's at least longer than 8 seconds of walking, but I just picked the longest one in that period. So if they walk, we can do it. And that's what our method is right now. Our error on that approach is about 4 degrees, which is exciting. We're pretty excited by that. But yeah, that's definitely been an issue, and I'm glad you brought up that question. But that's been an issue for actually doing this remotely and getting reliable data.
Just to jump in on that comment as well, you know, sensor to segment calibration methods, generally all it requires is that you have a known activity or posture that the person's in. And so really the advantage is coming from just the automation comes from removing the need to supervise that, and just, you know, the classification, the problem is solved by identifying the activity, right? So you could have a person, a lot of people will calibrate a leg worn sensor by having a person walk. Well, if you can identify walking data without having a sensor calibrated method, which, for example, gait bouts can be identified from signal magnitudes, then there is your activity that you would otherwise have prescribed for calibration purposes, and it's just available because people do these activities in their daily lives. So having methods that can identify calibration activities, although they're not doing them for calibration purposes, but if you have a method that can identify activities that are useful for calibration without having calibrated data, that's kind of, you know, how the method would potentially advance.
Thanks for that, Reid.
I guess, yeah, what comes to mind along that topic is, you know, we often hear that we need to design in flexibility of where the sensor is. And we, you know, I think Reid mentioned that, you know, conveniently, when you put a sensor on the chest, you also get the cardiac signal, which is really good. But what's the freedom of where to move that sensor, and how does it impact the algorithm? And I think that we're trying to find what that, you know, area is on the torso where we can get, you know, good cardiac signal, and also be able to do this automated calibration. And I think that you know, maybe Brett, you can comment on the approach and the flexibility that we potentially have with wear location.
Yeah, I mean, just as Reid and I have mentioned, basically what we're able to do is just correct for differences in how that sensor's placed. As long as it's fixed to a segment, we'll assume it's rigid. we'll say that as long as it's on that segment and we can identify these activities, then we can be very flexible as long as it's within the same, as long as it's moving together, right? If it's, you know, on one side of the chest or the other, we are able to handle that.
Yeah, I think that's a big benefit, because oftentimes we see that medical devices that are adhered to the body have very specific placements. And that becomes an issue when you have to, as you're a patient, you have to take it off and put it back on at the exact same location, and having some room to move it around, but still have high algorithm performance is definitely desirable.
And
Maybe
that's
just one.
actually, oh, sorry, go ahead, Ryan.
Sorry, Ben. I was just going to add one more small point on this topic. You know, I think one of the real benefits of adhesive sensors is that they, you know, to a certain extent, can't move once you place them on the body. Certainly, they can move, you know, as you change them out. But you know, there are other wearable technologies out there that in many cases are even more popular for these types of studies, where you're trying to get access to sort of high quality measures of gait and postural sway that you know, are not affixed to the skin, and so can move throughout the wear period by you know, maybe a subject bumps a device that's strapped to the body around the waist, and it sort of shifts where that sensor is a little bit. And so those, I think what's sort of nice about approaching calibration from a sort of data-driven perspective is that you can ostensibly update those calibrations over time. And so, you know, I think it gives you the opportunity to extract data even when things that you don't expect to happen. Whether that's like a participant placing an adhesive device back in a different location, which they absolutely do, or if it's somebody bumping a sensor as it gets twisted around a limb, and sort of is producing data that may make less sense. And so having these opportunities for more data-driven techniques that allow you to update things over time is, I think, a direction that makes a lot of sense for the field to go.
Yeah, thank you for that, Ryan, and everybody else. So the whole conversation about, you know, location of sensor and so forth is actually a nice segue into another question on like if you look like historically on how what we now call digital health technologies were developed. Back in the day, the more traditional medical devices, they usually came as a package, right? You had your sensor, you had your algorithms running on that, and maybe in more recent history, you had some kind of cloud infrastructure to access that. Nowadays, that is starting to split out a little more, where you have algorithm development happening, obviously using sensors for their validation purposes, but trying to become more agnostic to whatever device happens to be used to collect the data in the first place, and whatever location of that device was used to collect that data. So can you all comment on how you see that evolve, and especially focus, considering the topic of our journal club here, on the algorithm validation piece? Like, how do you try to factor in as early as possible how you're going to make sure that your algorithm is compatible with a wider range of sensor input than the one that you happen to use to run your early stage validation?
Yeah, so I'll comment on this one. So we've definitely have seen, right, some sensors look exactly the same and work as you would expect. As long as you're meeting base requirements like sample range and frequency, you can say, okay, these work. But we also have seen where maybe there's a little bit of aliasing we wouldn't expect it. So all that to say, we definitely have had to consider this. And from my experience, the solution is to basically not rely on things that are about how tall or how much power, the magnitude of a signal. And I don't mean like, you know, the vector magnitude, because obviously we like that. Um, but what I mean is, right, like how much amplitude is in that signal. That might change, right, depending on the sensor or how it's like, it even could be how it's attached to the body. But what really doesn't change is the shape. So if we can rely on features that are derived from the shape, right, for example, the what Reed went through, right, we're not looking at, okay, how tall is that peak from that wavelet transform signal, that we're looking at where it crosses through zero in that valley, right? So those shapes will be consistent across a different sensor, and then we can just and the sampling rate, as long as that's consistent, that's what sets our wavelet and scale. So we can keep that consistent. We know the shape's consistent, so we can work like that. That being said, though, we have seen more activity count driven measures sometimes do change. So maybe kind of lean away from those at certain times.
I think we also have learned that we can't say that we've developed a device agnostic algorithm without vetting each and every device that we want to use. So we have to do the diligence of going through and identifying, okay, this particular algorithm with this performance is going to be compatible with this source device, this hardware, and here's the data to show that. And so I think that I think we still firmly believe we're, if you say agnostic, it's still, you need to back it up with, and here is the data that, that we've collected. The other thing that I think we've been doing more recently, well, actually, we've been doing this for quite some time, is that when we're doing a data collection study, put in more than one sensor. So have the patient wear multiple sensors if there's real estate to do so, because it's like patient data collection is expensive. Might as well get as much data
Right.
from a variety of pieces of hardware as you can. And so we've seen very interesting results doing that. And I think my question around the different device placement, certainly from a cardiac standpoint, you see a lot of a lot of different responses, but accelerometer too, based on how people move and what their posture looks like.
Now, Melissa, you are saying validation studies, right? We're not putting as many sensors as possible on people in trials, though, right?
No, we want to learn as much as we can from development, right? So
Yeah.
when we're developing and doing data capture for algorithm development and performance. But yeah, then yeah, Brett's right. Like the least burdensome approach, and we will decide which sensor based on the requirements for each study. And so being able to have a selection of hardware that we can choose from and a selection of body locations, then we can work with the study teams in defining what metrics are important for your study, what's your patient population, and here's, the right device that will provide you the data that you want.
Yeah, and speaking of least burdensome approach, since the lines between medical devices and consumer technology are blurring, most people nowadays have at least an accelerometer in their pocket. Many of them have one on their wrist as well, right? So where it is appropriate, which is definitely not going to be all cases, like you pointed out, if you need, for instance, ECG measurements on the chest, you might as well do it there because it's a more controlled, you're going to be able to control the data more. You're going to have a narrower set of conditions to deal with. But that's not always the case, right? And in those scenarios, you may want to look at, can we rely on sensors that people already have so that there's no additional burden of using something else on top of that?
Yeah, I think that comes down to what's your goal. If your goal is just long-term surveillance and monitoring, you can definitely more lean on that because you're going to get the measurements when you can basically, right? So say it's a wrist sensor, it's your phone in your pocket. You're going to use a couple different sensors or techniques just to make sure the data is what you expect, and then you can like process it. Right, that's great for somebody that's just like looking to track their gait speed over the course of a year. What that's not really good for is a pharma company that wants to run a study and see if their drug is helping right their 95th percentile gait speed, because we're going to have very noisy measurements. We're not going to be able to take measurements as often, right? So say my phone's not in my pocket. Say it's in, I don't know, I left it on the table. Okay, well, I missed that. So it really depends on the context of use here as well. So I, I I definitely push that question back to like, what do you want to do it for?
Yeah, it's all about context of use, right? To your point.
Yeah.
I think a piece that maybe factors into both of those kind of points is around even within a given sensor, understanding how so many of these algorithms that are developed these days to translate measurements from sensors to things that are actually physiologically reasonable or clinically useful leverage some sort of AI or machine learning. Oftentimes that's very black boxy. And so it's not necessarily clear at the outset how changes, even small changes in sensor configuration or measurement properties impact those ultimately predictions of a clinically useful construct. And so even within consumer grade wearables, understanding how a firmware change from firmware 1.2 to 1.3 changes the resulting estimates of walking speed and when those firmware changes may happen in the context of a clinical trial. I think those are really challenging pieces that go along with, even not just a device transition, like going from, device A to device B, but even within a certain device, going from firmware A to firmware B, how those transitions matter in the context of your measurements. So something to keep in mind.
I do want to add a quick little anecdote there. We witnessed from a wrist-based, more consumer device, a firmware change. It was reporting apparently strides at first, and then a firmware change, and it was reporting steps, which I believe it was supposed to be reporting steps the whole time, but it went back and just doubled everything. So yeah, that's not great, but that's kind of to Ryan's point where sometimes we can't really control what's going on. So again, if it's a mission critical situation, maybe don't rely on just what people already have.
Maybe broaching in another topic with the group, like when we're thinking of, let's say, the validation path, and especially the analytical validation path of least resistance, what often happens is that you take whatever clinical standard currently exists, and since that's your reference measure, you try to replicate that, right? Which sets you up for trying to replicate in hospital, in clinic assessments in the home environment, which is not necessarily, I think we're all in agreement, the best approach. It is a approach. It is often, again, the path of least resistance, but it is not necessarily focusing on making sure that we critically think about what kind of combination of technology and algorithms and whatever outcome we're trying to measure in a home environment is going to be most valuable to that indication, to that group of patients, whatever it is, right? Can you all, like from your own experiences, comment on how you kind of try to walk that line? Because it is a balancing act, right? Like if you're planning to submit to FDA, you know that they're going to compare you to whatever is out there right now. So you always have to take that into consideration. But where is that line and how do you walk it?
I can jump in here. I mean, I like to think of the inertial sensor signals that we measure as themselves an indication of the motion. We usually, we see people move positionally. And so when we see joint angle data, for example, that translates more naturally to what we envision, right? Because we can interpret more easily a 90-degree knee angle, for example. But as soon as somebody talks about the acceleration of a foot, in terms of whatever meters per second squared, suddenly that's just not something that I readily have the sense, hardware to sense, right? But it's no less an index of how they're moving. And so I've just started using the inertial sensor signals themselves, the time series themselves, as a measure of the motion in the same way that we traditionally would have the joint angles. And in the same way that people will characterize time series of joint angles to determine differences in some intervention or between groups, we'll do the same things on the inertial sensor signals themselves. Now, in that case, sensor to segment calibration, as boring as that may be, becomes really important because those are now that's going to change the signals that you're measuring, and you would like to be able to attribute any differences to some intervention or a difference in group rather than a configuration error. But that just gives one example. I think I've said this before. I think that we underutilize the data that's available in these sensors, and there are things that are interpretable, like stride time and stride frequency, that can probably be calculated about anywhere. But there's different levels of information that come from the motion of different segments that probably indicate more globally important things. And we would do well, I think, to think about how to extract that information from the data that we're already collecting.
Great. I do want to kind of tag along with that. If you recall some of our older work we did, right? You're talking about gait here. For the most part, when you detect walking, when you see walking in a sensor, it's because people are walking. Obviously, we showed it was several years back that, right, if someone walks to the fridge, it's different than if they walk to their car. But again, you can kind of control that, like, oh, okay, I'll just look at longer walking bouts and I'm good to go. I think that kind of what Ryan presented shows that it's not necessarily true for all activities, right? Where we see just replicating what you do in the clinic for, say, postural sway is not necessarily the easiest. Whereas gait, I'm like, okay, it looks like walking. It is walking. Okay, this person has been walking for 39 seconds. Okay, that's walking, right? And then I can do these analysis. For postural sway or standing, right, which in the clinic is somebody's instructed to stand as still as they can, and right, the deviation there is okay, well, they're trying to stand as still as they can. How much do they actually move? What if I'm standing still but doing the dishes? What if I am playing with my kid? Right, and I think that's really what we saw in that last paper Ryan presented, was that people stand for different reasons. Obviously, people walk for different reasons, but that has different impacts on the accelerometer signal. And what we did in that paper was kind of just try to lean into some clustering and try to find what's useful. Like, okay, where can we go into here to actually figure out what's useful? And that was kind of more, right, to get closer to that lab environment to kind of control that. So, right, that's one technique we can do, is just look at what's try to get, like, basically you're combing through the noise. But then I think there's another method we could do, which we did not do in that paper. And that's just, we have to just consider things a little bit differently. We have some methods on the cardiac processing side, where we're not necessarily looking at like for a specific activity, but we're just approaching it differently. Obviously, you might monitor more, so it's right looking something more like an activity count, so you can kind of just gather it throughout the whole day rather than just for one specific activity. But again, that comes with trade-offs.
Just to add that a little bit, I think one of the ways that we've been trying to tackle this is really thinking deeply around what are the constructs that we're trying to measure, and making sure that we're validating sort of the core pieces of some of these algorithms that we know like need to line up. Like we need to know that the postural sway measures, for example, are actually measuring postural sway, but then embracing the fact that you know it's not entirely clear what we should be comparing at home, and sort of this idea that there are measures of capacity and there are measures of behavior patterns that we haven't been able to capture in the past, and so it really is kind of a blue sky territory where we could use these to derive potentially new measures of constructs that we hadn't previously thought about, but may be impactful for a specific health condition, and trying to kind of weave those together to really advance what you're able to leverage these these new data collection modalities to do. And I think one area that we've been working in is trying to understand sort of walking capacity in kids with DMD. And so I think everybody knows that the stride velocity 95th percentile measure that's sort of worked its way through the regulatory bodies. But I think there is a lot of unexplored territory there as you start to look at sort of free living movement and movement capacity and movement patterns, even within the context of DMD. We've had some recent work showing that postural transitions in that population may be even more sensitive to DMD related impairments than gait related measures. And that's something that hasn't really been characterized in the past in a free living way, but I think is looking at the patterns of those transitions and capacity to perform those transitions could be a really interesting area to continue to explore, but doesn't necessarily have a direct analog in the clinical side.
Alyssa, I saw you come off mute. Were you gonna add something here?
Yeah, I mean, I think like my first thought when you asked the question is that it's a bit of an unfair battle because it's in some of these cases it's apples to oranges. So like the analog, the legacy way that we measure things is just so different from what is possible with the digital sensors, but yet we're still kind of obliged to find that ground truth that we're comparing to. And so oftentimes the ground truth, or sorry, the gold standard is, it has flaws, and I find in some of our projects it's an uphill battle because um, we're trying to correlate a digital measure with a known limited primary endpoint. And we're never going to get there if that continues to be the mandate and it continues to be our approach. So I'm hopeful that with more data that we collect, we start to see the kind of the population, the start to understand better what this data looks like in mass populations so that we can kind of prove some points around the clinical efficacy of the data that we're collecting and not necessarily always have to fall back on comparing it to what was done, 20 years ago. And knowing that it adds a different dimension, right? And the way that we talk about it is that if you want to enhance your understanding of people and behavior and mobility, this is another view to look at that, to provide that 360 view of a person and what they're doing in the clinic and at home in a very, highly accurate way.
Yeah, absolutely. With that, we're coming up on time. So Ryan, Melissa, Brett, Reed, thank you so much for coming here to discuss your work and lending your expertise. Much appreciated. I have like a quick closeout here for everybody who's online about a new project that's going to be launching in September on core measures of mental health. So I alluded at the start of the conversation to the fact that we've completed this work on core measures of physical activity. If you look at mental health, a lot of assessments right now are very subjective. So we are bringing a team of organizations together that are kind of define the set of core measures in mental health that can be captured with sensor-based digital health technologies, which is a big one that we're really excited about. It is launching soon. So if you want any more information, the time is now to reach out to us over the website, because we are putting the final touches on the team of organizations and partners that is going to tackle that work. And with that, again, thank you so much to everyone who was here, everybody in the audience. I hope this was helpful to you, and hope to see you soon at another DiMe event. Thank you all so much.
Thank you, Ben. Thank
you.
you.
Thank you all.
Thanks.